Thanks everyone for joining this call. And I would like to thank the organizers of GNOBB for arranging such an amazing webinar. So briefly, I am Rashidul Islam. I have obtained my PhD in bioinformatics from the University of British Columbia, Vancouver, Canada. And I have got my BSc graduation from Genetic Engineering and Biotechnology at SUST. So today we will talk about genomic data and how we use different tools and techniques, including AI, to get the insight from the genomic data. So the genomic data or biological data is very complex. So it looks like the pieces of jigsaw puzzle. So at this point of time, we can generate different types of data. For example, using DNA sequencing technology, we can sequence the entire human genome. Also, we can sequence the epigenomes to identify the regulatory maps of the genome. Also, we can generate data of proteomics, metabolomes, and many others. From those data, we get the insight. So here I'm showing an example. By putting together the pieces, we can reconstruct the globe that has a meaning. And through analysis the genomic data, we can also get insight to understand the biology and the mechanisms. It is important to note that there are some missing pieces. Although we are good at generating in the data, it is very difficult to study human health and diseases. There will be always some missing gaps. And as long as we are adding more and more data, we can get better and better picture about our health. So there are some challenges. For example, interpreting the data to get the insight, there is a big challenge in there. Also, most of our genome is non-coding, and the regulatory part in the non-coding area is also challenging to study, and to link their function to the coding genes is a difficult task. Also, complex diseases like cancers, type 2 diabetes, cardiovascular diseases, all those diseases are influenced by many different factors. Here I'm seeing environmental factors, which are known or unknown. So there are also challenges to study those diseases. So since today we will primarily talk about genomic data, I wanna talk about the history of DNA sequencing technologies, how it evolved over time. In 1952, we came to know about the double helix structure of DNA. And later in 60s, Frederick Sanger proposed the Sanger sequencing method. After commercialization of this method, we sequenced smaller genomes, as well as it led to the foundation to sequence the Human Genome Project. In 2005, we have got the second generation or next generation sequencing technologies. Those are short read sequencing technologies, but high throughput and cost effective. That lead to generate an enormous amount of data after 2005. And right now we are living in the third generation sequencing technology, where the long read sequencing technologies are capable of sequencing larger fragments of the DNA, like several kilobases of DNA bases, and with higher accuracy. So that is helping us to explore the new biologies from the data. Here I'm showing an example how the sequencing cost drops over time. After 2005, there is a sharp drop of sequencing cost. This is an example for sequencing of human genome. In 2000, it took $100 million. Right now, we can sequence human genome around $500, and we are expecting it will drop to $100 in next few years. So after the invention of short read sequencing technologies, primarily from Illumina, the sequencing cost drops. So there is a correlation with the reduction of cost and the amount of data we are generating. In this plot, I'm showing the sequencing data that has been deposited into different databases, such as in GenBank, UniProt. I want to highlight this green line. This is the SRA database, the amount of data that have been submitted to SRA database. SRA people submit raw sequencing data to SRA database. So we can see after 2005, people are generating more and more data, and there is an exponential growth in the amount of data that have been submitted to SRA database. So today I will show some data on human genome. I would like to show how human genome look like and where we are at this stage. And this is the genome version 38. That was the previous version of the genome. And this is an example of chromosome 1. We can see in the chromosome 1, there are some missing pieces. There are some gaps in the genome. Those regions have repeat regions or complex genomic structures. We could not resolve those regions using short-read technology. Recently, we have filled all the gaps of the genome. Now we understand the entire human genome. Using the long-read technologies, we have solved these missing pieces. So this recent genome was published in 2022. This is called T to T genome, telomere to telomere, the one end of the chromosome to the other end of the chromosome. We know the entire genome. Here we are showing some examples, so sample 1, sample 2, and sample 3. We have enormous diversities and differences in DNA sequence within the population. For example, when we compare sample 1 to the reference, this individual has an extra copy of DNA that is not present in the reference, but this individual carries this extra copy. Also in the sample 2, this individual has some missing bases, and sample 3 has an insertion of certain DNA sequence when you compare to the reference. So those differences gives us the diversity and variation between the individuals. Often these variants, genomic variations, is also associated with certain diseases. So here is the summary of human genome. We have around 20,000 protein coding genes and around 25,000 non-coding genes. Non-coding genes influence the protein coding genes in the upstream or downstream processes. Also, we have variation in transcripts. Through alternative splicing, we get around 250,000 of different splicing variants of transcripts. So not only these protein coding genes, but also the non-coding genes, as well as splicing variation, gives the enormous diversity to make a complex network to function in the cell. So here I'm showing an example of two groups of individuals. One has the DNA sequence C. At the same location, another group is carrying the T bases. These are the difference. Let's consider this is a population with certain disease, affected cases. In affected cases, most of the individuals have T instead of C, whereas the healthy group contains the C base here. So when you perform a statistical association with the genetic variation and to certain phenotypes, we get a plot like this. The y-axis is the statistical significance, but this plot is telling that at the chromosome 3, this variation of C to T is significantly associated with this disease. So this is how we associate different genomic variants to certain diseases. And this is called genome-wide association studies. So here I'm showing one example of a BRCA mutation. That mutation is prevalent in the individuals with breast cancer. And this is a germline mutation. This causes the loss of function of BRCA mutation, BRCA gene that triggers the onset of cancers. So people carrying this BRCA mutation has 50% higher risk than the general population to have breast cancer at the age of 50. And at the age of 70, the risk increases up to 87%. Also, with the same variation, it increases the risk of ovarian cancer at the age of 70. So it is important to note that although individuals are carrying BRCA mutation, there are 50% of the individuals, they don't show the symptoms of the disease, or they never have the disease in their lifetime. Also, there is a factor, age factor. The people carrying the same mutation, but they don't get the cancer in the earlier stages of their life. In female, after menopause, there are changes in hormones and which may trigger the onset of cancer after the age of 50. So not only genetics, the presence of mutations is determining the cause of disease, but many other factors, for example, age or many other factors that we don't understand better, are contributing to cause this disease. As I said, most of our genome is non-coding. Only 2% have the coding exons. Rest of the genome is intron or intergenic regions. Those regions contain the regulatory elements of the genome. And in my PhD, I primarily studied the regulatory region or the non-coding region of the genome to understand its function and role to cause different diseases such as cancer. So to understand the regulatory regions of the genome, we need to understand the epigenomic technologies. In this example, we know that DNA is tightly packed on the nucleosomes, and this area is called euchromatic region because the DNA is accessible to the transcription factors to bind and initiate transcription. And those nucleosomes, the histone tails, can have different covalent modifications, such as acetylation, methylation, phosphorylation. Also, the DNA can also have CpG methylation, the cytosine methylation. So the presence of this covalent modification indicates the function of this DNA segment or the genomic segment. So these are called histone marks. The presence of certain modification indicates the function of this location. For example, if a histone 3 protein has acetylation at the 27 residue, this indicates this region has an enhancer. If it has K4 trimethylation modification, that indicates it's a promoter. So those modifications are used as a marker to identify the function of the genome. And this part of the genome is called heterochromatin, where this DNA is tightly packed to the nucleosomes, and those DNA are not accessible to the transcription factors to bind because they are very tightly packed. So there is no transcription in this location. So these are called transcription off and the heterochromatic regions. So heterochromatic histones also have certain marks. For example, trimethylation at the lysine 27 or canine trimethylation. If those modifications are present, we call them heterochromatic region where there is no transcription. So using chromatin immunoprecipitation sequencing or ChIP-seq, we can identify the genomic locations where there is a promoter or an enhancer or this region is heterochromatic. So there are many protocols to identify those locations. Here I'm showing one example of ChIP-seq protocol. We use antibodies to target those specific modification. Let's say this histone has an acetylation modification. And with antibodies, we target those modifications and pull down the nucleosome with DNA. Then we separate the DNA and followed by sequencing. When we map the sequencing reads to the genome, we find some peaks. The presence of a peak indicates this region of the genome has this specific modification, such as acetylation. So this technology allowed us to map the entire epigenome to understand the regulatory elements that are present in the genome. So here is an actively transcribing gene. This is the gene promoter, which is marked by K4-ME3. Polymerase 2 is binding there. Also, two enhancer regions are interacting distally to this promoter to enhance its expression. So how do we identify the enhancer locations? Using ChIP-seq, we can identify those regions that have acetylation mark, and those regions are the enhancer regions. So there is a global effort through International Human Epigenome Consortia. They generate those data to understand the regulatory map. They generate the data for K4-ME3, 27 acetylation, and many other histone modification marks, as well as DNA methylation, gene expression, whole genome sequencing. And here is Dr. Martin Hurst. He's my PhD supervisor. And Dr. Hurst is the chair for Scientific Steering Committee of IHEC. IHEC is generating data from different countries, and they are putting all the data together. Most of the data is publicly available. And so far, they have generated more than 7,000 data sets. from different tissues. Most of them are histone modification data. So the purpose of IHEC is to study human tissues, to understand the epigenomes and the transcription regulation. So in my PhD, I worked on the IHEC dataset. So I will show some data on my work. Before that, I want to give some context. So this picture on the left side is the classical epigenetic landscape proposed by Conrad Waddington, where a rolling ball have the potential to choose different paths during its downhill journey. In my PhD, I studied the hematopoietic development as well as their malignancies, such as the hematopoietic stem cells can choose different paths to develop into specific hematopoietic cells. And this hematopoietic development perfectly reflects the classical epigenetic landscape. I primarily focused on understanding the T cell and B cell leukemia. Today, I will show some data on B cell development as well as on B cell malignancies. So I studied chronic lymphocytic leukemia, which is a B cell leukemia. And also I collaborated on another project on follicular lymphoma. This is also a B cell malignancy. Both the diseases occur in the later stages of life, and those diseases are predominantly found in the European and American ancestries, and those both cancer have recurrent mutation on certain genes. So to understand CLL, we collected data from different B cell subsets. We need to understand the entire B cell maturation process. Then we will identify the epigenetic signatures that are specific to CLL. So B cell development is a stepwise process. For example, from hematopoietic multipotent progenitor cells, it develops into precursor B cell, the naive B cell. The naive B cell enters into germinal center. Through germinal center maturation, cells either exit as memory B cell or antibody secreting plasma B cells. So germinal center is an unique niche where global epigenetic reprogramming takes place, as well as cells acquire somatic hypermutations in the immunoglobulin genes. So the presence of somatic hypermutations is used to group CLL samples into two major groups. One carrying the somatic hypermutation, mutated CLL, one is unmutated. So this is a classical grouping, and UCLL is more progressive disease than MCLL. So to study all these cellular subtypes, we have collected data from IHEC. This is called reference epigenomic data that has all 6 histone marks, accessibility mark, and gene expression for like RNA-seq for gene expression, whole genome bisulfite for DNA methylation. This is a large amount of data. 50 reference epigenome is a big data, and this is one of the largest study in B cell and CLL. So we develop technologies and pipelines to analyze those data. So I'll show some results, how we process this data initially. We have identified the cell type specific or tissue specific enrichment of different histone marks. Then we group them and plotted in the two-dimensional plot using principal component analysis. This is an unsupervised clustering. So what we have observed that CLL forms distinct group for most of the histone marks compared to rest of the cell types. And a similar grouping was also reproduced in whole genome bisulfite data as well as RNA-seq data. So enhancers play a critical role in certain cancers to drive the overexpression of certain oncogenes or proto-oncogenes. In CLL, we have identified around 2,730 active enhancers that are specifically become active in CLL compared to rest of the B cells. Here, the blue color is indicating the presence of enhancers, and whitish is lack of enhancers. So in B cell subsets, we don't see those enhancers, but in CLL, they are active. For the same set of enhancers, active enhancers, I have plotted K4ME1, where these regions are marked by K4ME1 in CLL. Also, they are marked in B cells. So this is telling us, this mark is telling us that although those enhancers were present in B cells, but they were not active. They were poised. So they got activated in CLL very specifically. Also, these poised enhancers were heavily methylated in B cells, but undergo hypomethylation in CLL. So here I'm showing one example of BCL2 locus, where this BCL, this loci is enriched with H3K4ME1 across B cells and CLL. But in CLL, it has specific enrichment of H3K27 acetylation mark, but acetylation mark is absent in B cells. So this is how BCL gained 12-fold more higher expression in CLL compared to the B cells. Through the gain of acetylation mark, it is increasing or driving the overexpression of BCL2 genes. BCL2 is a known oncogene in CLL. So based on the data, we have proposed that enhancers that are poised or inactive during B cell development, but in CLL, they acquire acetylation modification and hypomethylation to become active. And through activation, it triggers the expression of certain genes that are well annotated in CLL to be oncogenic. And that involves the pathways such as immunoresponse, apoptosis, ONT, and JAK state signaling pathway. So later we want to classify the groups. This is very common in cancer research. We try to group the patients, whether they have different outcomes or not. And we have found that DNA methylation at the enhancers vary between the individuals. Then we performed unsupervised clustering to cluster the CLL cases into groups. There are two major groups. One has relatively lower methylation, another has relatively higher methylation at the enhancers. And those grouping is not dependent on the conventional UCLL to MCLL grouping. Also, we reproduced the same result in a different cohort of 105 CLL cases. It formed two distinct clusters, similar to the previous one. Also, it is independent of traditional classification. So here, we also looked into the survival data, and the patients with lower enhancer methylation has poor prognosis compared to high methylation group. So through this analysis, we have proposed a new method to group CLL cases, which is better than the conventional approach to classify based on IGHV mutation. And in another project, I studied single cell technologies to understand follicular lymphoma. Here, I'm showing an example that if we take a sample from a breast tumor, the sample may look like this. It has normal breast tissues that contains different healthy cells at different maturational or developmental stages. Also, it could contain the immunocells within the tumor. The tumor cells may have different proportions and different types. So without studying the tumor at a single cell level, we cannot get the better picture of what is inside this tumor. This could be very different between the individuals. The composition, the cellular composition could vary between these in individuals. So we need to understand the cellular composition to provide better therapeutics. Last 15 years, there is a significant progress in single cell studies. We can separate each of the cell and extract DNA from the single cell nucleus for DNA sequencing, as well as we can sequence the RNAs. Also, we can perform several epigenetic assays to understand the epigenome at the single cell level. So single cell technologies is now allowing us to understand more detail inside the tumors. So here we studied follicular lymphoma. We had 150 samples here in this two-dimensional UMAP plot. we have plotted all the cells from these 150 individuals with follicular lymphoma. And we used cell surface markers. The technology is called single cell cytof. Using this technology, we found two major groups. We have highlighted those patients in red and blue. And since although they are the same disease, but we have found patients have two types of cellular compositions in these two groups. Then we reproduced the same results using an orthogonal technology, which is single cell transcriptome analysis. And we found the similar results. For type A disease, you can see these 4 samples is clustering together to the germinal center B cells. They are very similar to germinal center B cells, whereas type B patients is further away from germinal center B cell, rather they are very close to naive and memory B cells. So these results is very similar to this previous analysis. From the cell surface markers, we also see the type A is more like GCBC, and type B is more similar to the naive or memory B cells. So we also showed that the type B is more aggressive type of disease compared to type A. So through analysis single cell, we can see although people have same disease, but they could originate from different cellular subtypes. Some could originate from germinal center, some could originate from early stage, such as naive B cells. So this technology is allowing us to understand the cellular composition, as well as to understand the tumor microenvironment better. So that is allowing us to target with precision therapeutics and develop future potential therapeutics. With single cell technologies and AI, right now there are many foundation models that are being developed to study single cell data. So single cell data is providing us enormous power to understand what is the difference between cell to cell and tissue to tissue. And because of the high resolution and the large volume of data, that is allowing these AI methods to tell us better about these differences and similarities, or the hidden patterns that are within the data. AI has another application, such as developing predictive models. In my undergrad thesis, we developed an AI predictive model that actually predicted the short human RNAs, which are called microRNAs. We trained the AI network with the existing known microRNA sequences. Then it was able to predict the unknown sequences, whether they are microRNAs or not. Also, during COVID pandemic, we developed another AI pipeline, virus Texo, to classify the taxonomies of virus sequences. After the pandemic, I thought, okay, we need to develop a method that can automatically assign the right taxonomy to the viruses. And these methods were trained on the known virus sequences, and that can predict the taxonomy based on the sequences from metagenomic data. This was also published in 2022. And this is part of my voluntary work in Omics Lab. I run this platform, Omics Lab, where we do voluntary work and generate bioinformatics pipelines. We learn from each other. I supervise Bangladeshi students through Omics Lab. When we find something meaningful, we try to publish them. So if anyone interested to join Omics Lab, please send me an email. I'm happy to work with you. Also, I am creating online contents on bioinformatics. I have released 2 complete courses on R programming, as well as on bash commands in Linux. These are the basic techniques we need to know before we start bioinformatic analysis. And also recently, I am uploading videos on 2 other courses on genomic data and file formats and bioinformatics tools and analysis. If you are interested to learn more, please check my YouTube videos. Finally, I would like to thank my lab members at UBC. And these are my funding sources from Search Network, Bioinformatics, Terryfox, UBC. Yeah, thank you very much for your time. I'm happy to take questions if you have any.